Hey everyone, it's Dr. Marcon, and this is Chapter 2, The Chemistry of Life. Student learning outcomes for this chapter include drawing atomic structures and being able to identify various types of bonding between atoms, describing and providing examples of the major macromolecules that living things are made of, also learning about being able to elaborate on the significance of the above molecules in life's functions. So in the last chapter, we talked about the properties of life. What is life? Well, building on that information, we know that life is made up of matter. And matter occupies space and has mass. It is composed of elements, and everything around you is matter. So next we're going to get into the building blocks of molecules. So even our food is made up of matter, and everything that has matter can be basically boiled down to different combinations of elements. I love coffee, and we know that more than 1,300 chemicals are within a cup of coffee. We know we have caffeine that makes up regular coffee, although some people do drink decaffeinated, which to me doesn't make sense. But for the most part, most cups of coffee contain caffeine, and caffeine is made up of different elements. So we can see carbon, hydrogen, nitrogen, as well as oxygen. So we have different numbers of these elements that combine to form caffeine. Okay, and these elements can be found in the periodic table. So just looking at that, how many elements are in caffeine? Well, first we need to define what elements are. So elements are substances that cannot be broken down or transformed into other substances. Elements are composed of atoms. Elements are designated by their chemical signal and possess unique properties. So there aren't many ingredients in humans or that make up you. We have the big four elements in your body. So the big four including oxygen. So the majority of our body is made up of oxygen, about 65%. And that makes sense because we need oxygen to allow for metabolic processes within our body. Carbon makes up about 18.5% of the body. Hydrogen, 9.5%, and nitrogen, about 3%. And the other 4% are a combination of these ingredients or elements. So we have calcium, phosphorus, potassium, sulfur, sodium, and chlorine. And we also have what's known as trace elements. So these are trace amounts, less than 0.1% of 15 other elements found in the body. So for example, we have iodine. Iodine is a trace element that is important in the formation of thyroid hormone. And iodine is required in small amounts. However, a deficiency in iodine will cause a pathological condition known as goiter. Goiter is the enlargement of the thyroid gland, and the thyroid gland can be found in the anterior portion of the neck. So goiter is a deficiency of iodine in the diet. If you look at your table salt, in most developed countries, we see that table salt, or those big packets of salt, they are iodized. So this allows for putting iodine in our diet so that we don't get conditions such as goiter. But again, these trace elements are only required in small amounts. They make up about less than 0.1% of the elements in your body. And here are some of the trace elements and their functions. So we have iron. Iron being very important for energy transfer by hemoglobin and other cytochromes. Zinc. Zinc is a trace element important for growth, healing, as well as immune function. Copper. Copper has connective tissue formation and energy transfer functions. Manganese. It's important for arginine, pyruvate, and superoxide metabolism. Chromium is part of the insulin receptor, so the receptor that binds with insulin to allow for transport of glucose into the tissues. Selenium. Selenium prevents peroxidation of reduced compounds such as glutathione. So these elements, although they are small, have very important metabolic functions within the body. So again, elements are chemically pure. These are substances made up of a single type of atom. Okay, and the periodic table of elements shows you the different elements that can be found. And if you'd like to use scoops of ice cream to help you visualize the elements, you can. So atoms are the smallest unit of an element that participates in chemical reactions. Okay, so elements are made up of a single type of atom, and atom being the smallest unit. And here we see an atom with the different components that make up the atom. Just like we have different flavors of ice cream, and we have different components that make up the ice cream. So how many different elements come together to make up caffeine? So we know that we have carbon, hydrogen, nitrogen, and oxygen. So that is 1, 2, 3, 4 elements that make up caffeine. But we can see that we have numerous atoms of each element that make up caffeine. And it's this number of elements that make up different molecules. So here we have 8 atoms of carbon, 10 of hydrogen, 4 of nitrogen, and 2 of oxygen. So now we get into the basics of atomic structure. An atom is made up of a central core called the nucleus. Within the nucleus, there are protons and neutrons. The proton is positively charged, while the neutrons have no net charge. Surrounding this nucleus are electrons. The electrons orbit around the nucleus at nearly the speed of light in energy shells. Now, electrons are negatively charged. So everyone, this YouTube link actually is a helpful 3-minute video that will help you kind of understand basic atomic structure. So I definitely recommend that you guys follow this link here that is on this slide. So this slide is a nice little 3-minute video on basic atomic structure. So when we look at an element in the periodic table of elements, we see that the element is represented by a letter, and then we have a bunch of numbers that kind of are found on the top and the bottom of the square where the element is found within the table. So what do these numbers mean? First, we know the letter represents the element. So in this case, O represents oxygen. The number above the element or the letter is the atomic number. Now the atomic number is equal to the number of protons. So we can see here oxygen has 8 protons, and the protons of course are located in the nucleus of the atom. Then there's a number below the element, and we can see that the number below is 16. 16 represents the atomic mass, because we know that matter has mass and occupies space. Here the atomic mass is equal to the number of protons plus the number of neutrons. And we can see that we can actually find out the exact number of protons just by subtracting the number of protons from this atomic mass. So 16 minus 8 means that there are 8 neutrons to this element. So atomic mass equals the number of protons plus neutrons. We know that there are 8 protons to oxygen, and just by doing simple calculations, simple math, we know that there are also 8 neutrons with this element of oxygen. So atomic mass for oxygen is 16, again being the number of protons plus the number of neutrons. So what about electrons? Well, we know that the number of electrons is actually equal to the number of protons. So if there are 8 protons to this element, that means there are 8 electrons. Now, electrons orbit the nucleus in what are known as shells. So electrons are arranged in energy shells. Going back to oxygen, we know that there are 8 protons. The number of protons is equal to the number of electrons. So we have 8 electrons in an element of oxygen. Now, the electrons follow what's known as the octet rule. Octet means group of 8. So the outermost shells of elements with low atomic numbers can hold 8 electrons. An element can donate, accept, or share electrons with other elements to fill its outer shell to satisfy this rule. So the innermost orbit of an element can only fill 2. So there are only 2 in the innermost orbit. However, the outer orbits have to fulfill that octet rule, meaning they have to have 8 to complete the orbit in the outer shells. So we know that oxygen has the atomic number of 8, so it has 8 electrons. So 2 will fill up the innermost shell, meaning there are 6 in the outermost shell. 2 plus 6 equals 8. Now that means that there are 2 more electrons that are needed to complete the octet rule for oxygen. So part of the rules, each shell must be filled before the next one is used, meaning this innermost shell has to be filled with 2 electrons before the next orbital can be filled. Other rules. So there are energy levels of electron rules. So again, the first energy level, or the first orbit, can only hold 2. The second shell can only hold, like the octet rule says, 8. And the third shell can also hold up to 8. So again, this is only for the first few elements of the periodic table, and this goes up to calcium, which has an atomic number of 20. However, what's important to note is the majority of important elements that you as a biology student will deal with are within that range. So we're not going to be looking at higher elements with larger atomic numbers. We're only looking probably up to calcium. Again, calcium has an atomic number of 20. Now, an atom's electrons in the most outer shell determine how and if the atom will bond with other atoms. Outer shells that are not full will tend to react with other atoms. So given what you've learned so far, which of the following atoms are most likely to react with or bond with another atom? So first you want to start drawing the atomic structure of each atom below. So we know that carbon has the atomic number of 8. So if there are 8 protons, that means there are an equal number of electrons. So in this case, carbon with the atomic number of 8 will have 8 protons and 8 electrons. Helium, having an atomic number of 2, again, atomic number represents the number of protons, and the number of protons equals the number of electrons, so the number of electrons for helium is also equal to 2. So which one is more likely to react with another atom? So let's start by drawing the atomic structure. So we looked at carbon. We know that carbon has an atomic number of 8. And the number of protons equals the number of electrons. So that means there are 8 electrons to carbon. So first we draw the outer shell, or the innermost shell, excuse me. So the innermost shell can only fill up to 2 electrons. So we can draw those electrons in the innermost shell there and there. And then the remaining we have to draw in the next shell. And we know that the next shell can hold up to 8 electrons. So we've already used 2 of the 8, so that means we have 6 electrons remaining. So we can draw it within this orbital here. So let's draw 2 there. We can draw 2 there, however you want to draw it. But no, we have to represent 6 electrons. Okay, so here we have our 6 electrons. So that is how we would draw the atomic structure of carbon. Then we have helium. Helium has an atomic number of 2 protons, so that means there are 2 electrons. So let's draw helium here. Oh, my drawing seems to have disappeared. Let's go back. So back to carbon. All right, guys, I am not an artist. So again, we have 2 in the innermost, and then 6 in the next orbital. And then we have helium that has atomic number of 2, so 2 protons, meaning there are 2 electrons. And the first orbital, we can say, is full because it has 2 electrons exactly. Now, which one of these would be more active, or would be more likely to bond with another atom? Would it be one that already has all its orbitals full, or would it be the element who still has missing electrons in the outermost orbital? So carbon is more likely to form bonds with other atoms. So again, an atom's electrons in the outer and the most outer shell determine how and if atoms will bond with other atoms. And then again, outer shells that are not full tend to react. Make sure you guys check out this YouTube video here that goes over basic atomic structure as well as the octet rule. So what happens when atoms interact with each other? You will get structures known as molecules as well as compounds. So here we can see a single atom, yum, chocolate ice cream, and we have different flavors just like we have different elements within the periodic table. So molecules and compounds are products of chemical bonding. And chemical bonds are attractive forces that hold atoms together. So we can see in our different scoops of ice cream there are different bonds that will allow for the coming together of different atoms. Okay, so we can think of individual ice cream flavors as single atoms, and these atoms make up the different elements, and these atoms are held together by chemical bonds. So what bonds can you think of that hold atoms together? Here we can see just different pictures of different bonds, bond of marriage, puzzle pieces fitting together, and so forth. So atoms can actually interact in multiple ways. The first bond that we're going to talk about is an ionic bond. An ionic bond is a strong bond and it's formed between oppositely charged ions. So we'll have one ion that's positively charged and another ion that is negatively charged. A common example we like to use is salt. Salt is made up by 2 atoms or 2 elements, sodium and chlorine. So the sodium ion is actually positively charged, whereas the chloride ion is negatively charged. we can see that the sodium atom in its outermost shell only has one electron. Now, would this outer shell more likely donate an electron or accept 8 electrons? So more than likely it's going to donate its 1 electron. And it's going to donate its 1 electron to the chlorine atom, because the chlorine atom actually has 1, 2, 3, 4, 5, 6, 7 electrons in its outermost shell. So it is more likely going to accept 1 electron as opposed to donating 7 electrons. So this actually forms a very strong bond between the two atoms called an ionic bond. The sodium ion, the positively charged sodium ion, will donate one of its electrons to the negatively charged chloride ion, forming sodium chloride, also known as salt or table salt. The next type of bond that we look at is a covalent bond. Now, the covalent bond is a very strong bond, and this is due to electrons being shared between atoms. Examples of covalent bonds where we see sharing of atoms include methane, where we see the structure is CH4, so we have one atom of carbon and then 4 of hydrogen. And of course, water. Water is an example of a covalent bond, where we have two molecules of hydrogen and 1 of oxygen. Now, sharing or covalent bonds can be done one of 2 ways. Which one of these 2 pictures would you say represents a covalent bond where electrons are being shared equally or unequally? So here we see two cute little boys sharing a donut, whereas here we see typical siblings kind of pushing and pulling a stuffed animal. So we could actually say that this first picture represents an equal sharing of electrons, and this an unequal sharing of electrons. So equal sharing, unequal sharing. When we have an equal sharing of electrons, this is known as a non-polar covalent bond. Okay? When we have an unequal sharing of electrons, this is called a polar covalent bond. So here we can see equal sharing between 2 atoms of oxygen, and here we see unequal sharing of electrons between oxygen and hydrogen. So nonpolar covalent bond between 2 atoms of oxygen, polar covalent bond between a hydrogen atom and an oxygen atom. So polarity just describes how atoms share electrons. In this case, the donut, in this case, the stuffed toy. So why do atoms share differently? Some atoms, like bigger ones, are more attracted to electrons in a covalent bond than other atoms. So we can say that an atom is more electronegative than the other. Now when this happens, this can cause there to be an uneven distribution of charge within the molecule. So in a polar molecule, we have an uneven distribution of charge, whereas in a nonpolar molecule, there will be an equal distribution of charge, an equal sharing of charges. Okay, so that is the difference between a polar and nonpolar covalent bonds. So we can see that oxygen is pulling on the shared electrons and is a little more negative than hydrogen, because we know electrons are negative. So it's going to be more negative than hydrogen. The next type of bond that we're going to talk about are hydrogen bonds. Now, hydrogen bonds have weak attractions and usually occur between a partially positive hydrogen atom and another atom with a partial negative charge, such as oxygen. So these hydrogen bonds we can see with a molecule of water. We can see that we have the partially positive hydrogen and a partially negative oxygen molecule. And these in turn will create other bonds with other water molecules, creating larger water molecules. So take this slide to identify and justify the bond type in each of the following examples. So if we have carbon and oxygen that bind together in CO2, these share their electrons equally. What type of bond would that be? Would that be a polar covalent bond or a non-polar covalent bond? Water molecules stick together due to the partial positive charge of hydrogen in one water molecule, which attracts a partially negative oxygen from a neighboring molecule. What kind of weak charge is that? Or weak bond is that? Calcium will donate an electron to chlorine to form calcium chloride. What kind of bonds occur when we have bonds where electrons are donated? So try to figure out the types of bonds within this slide. So what are living things made of? We talked about properties of life. Now we want to know what they're made of and how are these structures built? How do we go from these atoms and molecules to a living, breathing person? So structure can be defined as what something is made of. So we have main types of molecules that all living things are made of, and we know we are what we eat. We are made up of inorganic molecules such as water and salts. And we are also made up of organic or carbon-based molecules. And these important molecules that make us who we are, that make us living and breathing, these are known as our macromolecules. Now, take note of the word organic. For me, the word organic is just a fancy title that companies like to use to drive up the prices of food. Organic literally means carbon-based. So we are all carbon-based. So we are all organic. So labeling something as organic, one, is redundant because everything is made, or that is made up of carbon is organic. So we have our carbon-based or organic macromolecules. We have we have carbohydrates made up of sugars and starches. We have our lipids known as fats. We have our proteins, and we have our nucleic acids, such as DNA, deoxyribonucleic acids, as well as RNA, ribonucleic acid. So let's talk about these macromolecules. We know that many small molecules will connect to form large molecules. These smaller molecules are known as monomers. So monomers are small molecules or building blocks for the larger molecules or macromolecules. Polymers, poly means many, are large molecules made of many similar or identical building blocks. So here we actually see smaller molecules come together through a dehydration reaction, meaning water goes out of the equation, forming a larger polymer. So we have smaller molecules. We have a short polymer and a monomer coming together and then forming polymers. So if you were going to build a large molecule, it needs to be strong. What type of chemical bond do you think works best for building polymers? Do we want the strong covalent bonds? Do we want ionic bonds? Do we want the weaker hydrogen bonds? Of course, we would want covalent bonds. The first macromolecule that we're going to talk about are carbohydrates. Carbohydrates are an immediate source of energy. They are a type of storage material that we could use should we find ourselves in fits of starvation. Also, carbohydrates provide the material for building structures such as DNA and RNA. This little cartoon is kind of funny. While he couldn't be sure this was the killer, Inspector Lynch knew now that he was dealing with a very complex carbohydrate. So complex carbohydrates, we think of our carbs, our bread, our pastas, and such. So complex carbohydrates are made up of smaller molecules. The most basic of the molecules are the monosaccharides. So carbohydrate monomers are monosaccharides. These include glucose, fructose, galactose, and ribose. These are our simple sugars. Now note, in order for the body to absorb our polysaccharides or our carbohydrates, they have to be broken down into the smaller, most simplest monomer or simplest form, which are our monosaccharides. We have dimers, or combinations of monosaccharides. When we have two, we have disaccharides. So disaccharides are basically two monosaccharides coming together and bonding. So we have 1, 2. So one monosaccharide here, another monosaccharide here, coming together to form a bond, and we have a disaccharide. Now, what is the bond that helps bring together these two molecules? That is a covalent bond. Again, we need a strong bond to bring these two molecules together. So examples of disaccharides include sucrose, our table sugar, lactose that we find in dairy products such as milk, and maltose. Now, I've included in this PowerPoint a table of the different carbohydrates. We have our disaccharides and polysaccharides and the monosaccharides that make them up. So, for example, if we have the monosaccharide glucose and the monosaccharide galactose come together, this forms the disaccharide lactose. And we'll talk about lactose in just a little bit when we talk about lactose intolerance. Basically, we have enzymes. Again, enzymes being a protein, we'll talk about proteins in a little bit, that basically break up this disaccharide into its smaller components, its smaller monosaccharides. So lactase, anything that ends in ase is an enzyme, will break up lactose to form the monosaccharides glucose and galactose, and so forth. So large molecules of carbohydrates, these are called polysaccharides. Poly means many. So here we have two disaccharides. Example of disaccharides include sucrose, lactose, and maltose. And then we have many of those disaccharides or monosaccharides coming together to form a polysaccharide. So examples of polysaccharides include starch, glycogen. Glycogen is the stored form of glucose. It's for energy reserves. And we have our dietary fibers such as cellulose. So all of these are made up of polysaccharides, which can be further broken down into its smaller components, its disaccharides as well as monosaccharides. So moving forward, we then go to proteins. Proteins are the molecular machines of living things. We can find proteins. We can find structural proteins, storage proteins, contractile proteins, as well as transport proteins. And we just talked about enzymes. Enzymes are proteins too. Now, the basic building blocks of proteins, or the protein monomers, are amino acids. And amino acids actually come in 20 different flavors. So the basic structure of an amino acid is an amine group plus a variable group and a carboxyl group. Okay? So this makes up an amino acid. So many amino acids form polypeptides. So a protein polymer is known as a polypeptide, and we can see that there are covalent bonds between the amino acids. Now we can go from different structures to more complex structures that form proteins. So here we have a long strand of amino acids bonded by covalent bonds, and here we see a more complex structure. Because protein function actually depends on structure. We can talk about primary structure, secondary structure, tertiary structure, as well as quaternary structure. And it all depends on going from sort of a long chain of amino acids to these chains all of a sudden folding in of itself, forming folds and pleats and becoming a more complex structure. And that structure helps determine the function of the protein. So, for example, how does the structure of these cars relate to their function? We can take a minivan, a simple minivan. We know that it's large, it has a large seating capacity, it can hold children, and its function is to transport people, to transport a family. Whereas, if we look at this middle-aged nightmare, this sports car, it doesn't really have that much of a seating capacity. It maybe fits 2 people comfortably, and this is more for showing off, more for racing around, driving really fast, and having fun. So you can definitely see how the structure of these cars will relate to their function. This car, not so much for driving around a family. So, with regards to primary structure, primary protein structure is made up of a chain, a long chain of amino acids. When we get to a secondary protein structure, this is a folding pattern of these chains. The tertiary structure will actually involve folding 3-dimensionally. And then with quaternary protein structure, there's more than one amino acid chain. Now, protein structure can be damaged. It goes through a process of denaturation. So denaturation occurs, is the process where proteins can lose their structure. So what types of conditions can denature proteins? We can have extreme temperature. We can have change in pH from either basic to acidic or acidic to basic. Also, exposure to specific chemicals can cause denaturation. So, for example, we have egg whites. We know that egg whites contain a lot of protein. However, when we whip them or use a hand mixer to beat them, we can see that they go from this sort of clear, almost clear fluid to this opaque, stiff texture. So basically, we are changing their structure by mechanically beating and changing the structure of the protein to form nice white peaks. We can either make a meringue or make a nice mousse by introducing air into the proteins to use for different methods of baking and such. Now, enzymes, we talked about, are a special type of protein. These help make reactions go faster. what type of enzymes do we know about? Well, we talked about lactase in a couple slides ago. So lactase is an enzyme that takes the disaccharide lactose and breaks it down into the smaller monosaccharides, glucose and galactose. So when we add water to lactose in the presence of the enzyme lactase, we will then get the monosaccharides, glucose and galactose, and we can see the OH will bond to glucose, whereas the hydrogen from water will bind to galactose. So this reaction, this is, because we're adding water, this is called a hydration reaction. We saw a dehydration reaction earlier where we took out water. Here we are adding water. So this is a hydration reaction in the presence of lactase, breaking down lactose into its monosaccharides, glucose and galactose. Now, activation energy is the amount of energy necessary to start the reaction. So when we add the enzyme lactase, we speed up this reaction of the breakdown of lactose into its monosaccharides. So basically, this activation energy we can compare to the energy needed to get up off the couch, or get up and walk away from our computer. This is activation energy. And once we get going, hopefully we don't stop. Now, enzymes are really sensitive. They're very specific. They work best at specific temperatures as well as pH. If the shape changes due to pH or temperature, they may not be able to catalyze the reaction. Again, they're very specific to where they bind to the substrate. Okay, so it has to fit exactly to its substrate. Now, an enzyme can be used again and again. So here we have an enzyme. If we add temperature, we can change the conformation of the binding sites, and this will allow it to bind to its substrate. So now what happens when you drink milk? Again, we have the enzyme lactase, which allows for the hydration reaction of lactose to be broken down into its monosaccharides, galactose, and glucose. So these lactase enzymes are found in the brush borders of your intestines and allowing for you to break down lactose. However, if you are lactose intolerant, which we'll talk about, so why do some adults get sick when they drink milk? If you are lactose intolerant, you basically no longer have that lactase enzyme in your intestines. And this can occur at any stage of life. For most human beings, we eventually all become lactose intolerant because we lose that lactase enzyme. Some different ethnic groups become lactose intolerant fairly early, some, right after birth. That's why we have symptoms of lactose intolerant. Basically, we can't break down the disaccharide lactose. So what happens is the lactose will sit in our digestive system, in our intestines, and we know that lactose is a sugar. And who likes sugar best? Bacteria. Bacteria in our intestines actually feed on the lactose that doesn't get broken down and exhibits symptoms of lactose intolerance, such as gassiness. Sometimes we can either have constipation or diarrhea depending on the symptom. So it's the bacteria that cause the symptoms of lactose intolerance because that lactose has not been broken down due to the deficiency of the lactase enzyme in our intestines. So moving on, next we'll talk about nucleic acids. So what do nucleic acids do? So DNA provides the instructions for making what? Provides the instructions for making us, living organisms. RNA provides the instructions for making proteins. So proteins are what direct most of life's processes. So we have genetic information, instructions for making living organisms, and then we have RNA, which provides instructions for making proteins. And again, these proteins are important because they direct our life's processes. So the monomers, or basic building blocks of nucleic acids, are nucleotides. And we have three components of our nucleotides. We have our nitrogenous base here. We have our 5-carbon sugar. If it's DNA, it would be deoxyribose. If it is RNA, it would be just ribose. And then we have a phosphate group. Oops, let me erase that. Erase. My eraser is not working, sorry guys. So again, we have our nitrogenous base, we have our 5-carbon sugar, and we have our phosphate group. So that makes up the nucleotides. So each nitrogenous base is attached to a sugar molecule, which is attached to a phosphate group. And here we can see that structure. Nitrogenous base, sugar, phosphate. So that makes up a nucleotide. Oops. Turn that off. All right. So again, we see our nitrogenous base, deoxyribose sugar, our 5-carbon sugar, and our phosphate. So how could we create a nucleic acid polymer? What bonds are we going to need? We also know that anything living contains nucleic acid. So DNA stands for deoxyribonucleic acid, whereas RNA stands for ribonucleic acid. So again, it depends upon the 5-carbon sugar. So we can see the polynucleotide. Here we have DNA. Again, we have the nitrogenous base with the 5-carbon sugar and the phosphate backbone, and these are they are bonded to each other. So we know that DNA has the characteristic structure of a double helix. So DNA is a double helical structure composed of 2 strands of polymers of nucleides. Now these strands are formed with bonds between the phosphate group and the sugar group of adjacent nucleotides. So here we can see a sugar group bonded to the phosphate group of the next nucleotide. So these are actually bonded together with hydrogen bonds. Now the strands will coil about each other along their length, giving the double helix appearance. So we can see it kind of coils around each other, these 2 strands coiling around each other, creating a double helix. So we know that these are important for storing and transmitting information. Next macromolecule we're going to talk about are lipids, or fats. So this is the last one we're going to talk about. Lipids are very diverse, but they do have some things in common. The one thing that they all have in common is they do not dissolve in water, and this is known as being hydrophobic. And most cats do hate water, although I did take a virtual field trip with my boyfriend's 9-year-old daughter, and we went to the San Diego Zoo, and there is actually a type of cat that loves to swim in water. So not all cats hate water, but most of them do. So lipids, we can characterize them or categorize them into fats and oils, also known as triglycerides. We have our phospholipids, and we have our steroids. So again, lipids do not like water. They do not dissolve in water, also known as being hydrophobic. So triglycerides are a type of lipid. The fats in the food that we eat like bacon, are called triglycerides. So it is made up of a glycerol molecule, which makes up the head of the region, and 3 fatty acids, and these make up the tails. So tri, meaning 3, so we have 1 glycerol and 3 fatty acids, and lots of chemical bonds between them. So fats are composed primarily of 3 elements. We have a carbon, a hydrogen, and an oxygen. And we know that energy is stored in many of these carbon-hydrogen bonds. So we use fats for energy as well. So we can see actually the symbol for fat or triglycerides. So we have that 1 glycerol molecule with the 3 fatty acids. And again, it's hydrophobic, does not like water, does not dissolve in water. Another type of lipid are steroids. Steroids have a ring structure. They have 4 linked carbon rings, so 1, 2, 3, 4, and several will have a short tail, okay? We know that cholesterol is a steroid. Cholesterol is mainly synthesized in the liver and is the precursor, meaning we need cholesterol to make many of the steroid hormones such as testosterone and estrogen. So testosterone in males, estrogen predominantly in females. And again, hydrophobic. Phospholipid is also a type of lipid. Phospholipids are important because they form the cell membrane. When we talk about cells, we'll talk about that phospholipid bilayer that makes up the cell membrane of the cell. Phospholipids are similar to triglycerides. They have that hydrophilic or water-loving head and the hydrophobic or water-faring tails. So here we can see the structure of a phospholipid, which is hydrophilic head and hydrophobic tails. Again, does not dissolve in water. And when we look at chapter 2, we'll look at the components, the phospholipids that make up the plasma membrane, and we can see it is made up of 2 layers of phospholipids, which the outer layer being the polar head or water-loving head of the phospholipid, and the nonpolar or hydrophobic tail of the phospholipid. And again, there are 2 layers to the plasma membrane of phospholipids, again making up the membrane of the cell. So the polar head is hydrophilic, where the nonpolar tail is hydrophobic. And that's it, you guys, for chapter 2 with regards to chemicals. It's not going to get too much more involved with that, but make sure you are reading chapter 2 on the chemistry of life.